Faustino from the Office of Pharmaceutical Quality, and this morning I'll be speaking to you about bioanalytical methods and validation, history, process, and regulatory perspectives. This presentation reflects the views of the author, and it should not be construed to represent FDA's views or policy. So in this first workshop session, we're going to focus on guidance and laboratory perspectives. You've already heard about the first talk. Our second presentation will be Regulated Bioanalysis for Large Molecules by Dr. Jinhui Zhang. And our third presentation will be Regulated Bioanalysis for Small Molecules by Dr. Dia Shaklia. At the end of that, we'll have a panel discussion, science-based perspectives for the application of the Bioanalytical Method Validation guidance. Our session objectives today: participants will learn about the evolving process of the Bioanalytical Method Validation guidance, and will understand the regulatory scope and the importance of bioanalysis. They'll learn about the FDA regulatory science perspective for bioanalysis, and participants will learn why FDA conducts bioanalytical studies and evaluates innovative technologies. Our presentation objectives: I want to give you a context for bioanalysis. And this next slide will help us to do that. I want to explain the Bioanalytical Method Validation guidance, its history, its process, its procedures. I hope to provide a foundational understanding of the importance of bioanalytical method validation. And I hope that we'll be able to provide information that can support you sponsors and researchers. So this was a slide that was presented by Dr. DeCilia at the AAPS 2018 Bioanalytical Method Validation Workshop. And it was a slide that was taken from a paper that was published a few years earlier. As you can see in this paper, bioanalytical validation is foundational. So any of the information that you would receive from clinical pharmacology from these studies, or safety decisions that you would make based on safety and efficacy, are based on the bioanalytical method validation, and that the bioanalytical method will do what it is intended to do. My presentation will be broken into three parts: a history of the guidance, its scope and its evolution, FDA bioanalytical studies from a regulatory science perspective, and bioanalytics. And we'll have a short overview of innovation, and we're going to discuss existing and emerging bioanalytical technologies. So I want to start with first principles. What is a guidance? So on March 7th, 1996, it was published in the Federal Registry of what an actual guidance is. And they are documents prepared for FDA staff and applicants related to the process, content, and evaluation of applications relating to the design and testing of regulatory products. They are guidances describe the agency's current thinking on a topic and are viewed only as a recommendation. It's important to understand that FDA guidances do not establish legally enforceable responsibilities, but they are a guide to you through the process and the procedures that you would use to develop and implement bioanalytical methods. So what is validation? Well, this document that you see on the right, submitting samples and data for analytical method validation, is from a 1986 guidance document. And this guidance document is really the document, it's a step off to the 1998 Bioanalytical Method Validation guidance draft. And so the language in this document says that method validation is the process of demonstrating that analytical procedures are suited for their intended purpose, and that regulatory method validation is the process of demonstrating that analytical procedures submitted in an NDA, a new drug application, or an ANDA are suitable for their intended use. The 1998 draft guidance has language that states analytical method validation includes all the procedures recommended to demonstrate that a particular method is reliable and reproducible. And 20 years later, in the 2018 document, they used the language of bioanalytical method validation, stating that bioanalytical method validation proves that the optimized method is suited to the analysis of the study samples. So I wanted to step now to the history of the guidance. We have a definition of what a guidance is. And in 1998, this draft guidance for bioanalytical method validation was based primarily on the workshops, analytical method validation, bioavailability, bioequivalence, and pharmacokinetic studies held in 1990 at Crystal City, and we will call this Crystal City 1. The meeting was sponsored by the American Association of Pharmaceutical Scientists, FDA, and a series of other national and international agencies. The guidance was prepared by the CDER Biopharmaceutics Coordinating Committee and the Clinical Pharmacology section of the Medical Policy Coordinating Committee in CDER, and staff from CDER's Laboratory of Clinical Pharmacology, where I actually started out when I joined the agency. There was a follow-up workshop in 2000. And this workshop was entitled Bioanalytical Methods Validation, a Revisit with a Decade of Progress. And as I talk a little bit more about these workshops and the workshop reports, I want, this report here is pivotal. Of the 5 workshop reports that I'm going to introduce you to, this particular workshop report was very important as the guidance was eventually published. The draft guidance was eventually published in 2001. So many of the issues that we discussed back at that 2000 conference in terms of partial, full validation, bridging studies, calibration models, spiked or incurred samples, or new technologies such as LC to LCMS, and this thing called ion suppression, this thing called matrix effect, which we're still dealing with today. And I want, as we talk about the guidance, technology has helped to advance some of the thinking and the language in the guidance. And back in 1990, probably 90+% of all clinical studies that were submitted to the agency were liquid chromatography. By 2000, probably 90% of the studies were LCMS. And by 2010, it was 95% plus that were LC-MS-MS. And so you can see not only the evolution of the guidance, you'll see as we chat a little bit about evolution of thinking, but also the technology has evolved. And as we look back at this 1998 draft, and we talk about the fact that bioanalytical method validation is a foundational document, a foundational guidance, these 9 or 10 references here really helped to form the basis of this first draft in the 2001 draft of guidance. And so for anyone who is really interested in seeing the origin and the basis for a lot of the language that's in these guidances, I'd recommend that you go back, take a look at my slides, take a look at these references. I want to take you now through a little bit of the history of the guidance, through Crystal City 1 through Crystal City 5, and some of the pivotal events. One of the things is, as we go through these next 2 slides, is these are a snapshot. So many other things have happened during this time, but these really are a snapshot of overall the development of the guidance and to give you a flavor of what's going on. So there was that Crystal City conference in Crystal City in 1990. As we talked about, publication in the Federal Registry that defined regulatory guidances, the draft guidance in 1998, Crystal City 2 in 2000. There was a follow-up workshop on macromolecules at Crystal City. There was Crystal City 3 in 2000. There was Crystal City 4 in 2008 that talked about current topics in GLP bioanalysis, assay reproducibility for incurred samples. There was Crystal City 5 in 2013. And importantly, the publication of the guidance in May of 2018. As we look at these events, as we put these together on one slide, you can see Crystal City 1 2, 3, 4, and 5. And in each of these conferences, there was conference reports. And so to understand the guidance and to understand the development and the evolution of the guidance, I think it's very important to go back and look at these conference workshops. They are illuminating. They talk about the issues of the day. And as we talked about earlier on, some of those issues, such as matrix effect that we were talking about back in 1998 and 2000, are with us in 2018 and 2020, which leads us to the conference report on International Conference of Harmonization, Bioanalytical Method Validation conference report from that June conference of 2019. And this report really is very important because this is how the FDA's domestic guidance is evolving into the ICH guidance that will be used in the major regulatory regions in the world. This is a snapshot of conference report 2, the one that I had mentioned. And these reports are really broken out, and they're pretty standard in terms of purpose of the report and objectives. And it'll talk about the issues and these ongoing issues. And so if you want to understand how the guidance is developed, how it's evolved, what were the important issues then and now, please take a look at these conference reports, because they have helped me as a researcher in the lab, and I believe that they'll help you as a researcher or as a sponsor. The scope of the guidance back in 1998 was very focused. It was clinical studies for bioavailability and bioequivalence and PK studies, and applications for investigational new drugs, NDAs, and ANDAs. And it may apply, as the language said, to veterinary drugs. 20 years later, the language has expanded, and the guidance has expanded to talk about non-clinical studies for bioavailability and bioequivalence, investigational new animal drugs NDAs, and ANDAs. And the guidance is really still broken out into two parts. It applies to chromographic procedures, typically column chromatography procedures, and ligand binding assays that determine the level of drugs and metabolites, and increasingly biomarkers. So in process procedures, as we look back at the guidance, and 20 years later to the 2018 guidance, we now have a whole section on method development. And that method development section really was part of what we used to call pre-validation. And this method development section, there is a process in there. It helps you as a sponsor or a researcher, the things that you should be doing in method development and documenting. And then method validation, the things that we would validate, those particular validation characteristics, accuracy, precision, specificity. And it provides very clear language on stability, those 6 stability studies for plasma stability or on-injector stability, and it talks about procedures. And here you see the evolution from HPLC or LC all the way to HPLC MS, which is the primary procedure that most people use, although people use GCMS and advanced bioanalytics, which we'll touch on a little bit later, are rapidly gaining use. And there's another important point from this particular guidance is fit for purpose. And so the guidance defines this fit for purpose. So will it support a regulatory decision, or will it be used in a research setting or a screening setting? And so there are 4 levels for fit for purpose. But if there's a regulatory decision that will be made, then that method must be fully validated. So as we continue on the evolution of the guidance, and we see the 1998 guidance, and then the 2001 draft, the 2013 guidance, and the 2018 guidance, which most researchers and sponsors are using. As this guidance evolves, it will evolve into ICH M10, Bioanalytical Method Validation. But in the meantime, the agency is still dealing with issues, and so it has published a guidance for industry on the evaluation of internal standard responses and issues in terms of internal standard variability. The next part of our study is FDA Bioanalytical Studies, a regulatory science perspective. Who does bioanalytical studies and how and why. So what is regulatory science? And so I've put a couple examples up here, repackaging and split tablets. And I want you to kind of keep those in mind as we talk about this. Regulatory science is a scientific discipline consisting of the development and application of scientific methods, tools, and approaches that can be used to support regulatory or other policy objectives. Regulatory research is focused on protecting and promoting public health, especially identifying emerging technologies, speeding innovation to support review or assessment, policy and compliance, and increasingly surveillance needs and requirements. So for repackaging studies or for split tablet studies, those are studies that industry would probably not do and academia would not be funded for, but the agency would do the regulatory science studies to help protect the public health, because these are things that happen millions and millions of times a day. And guidances were published to support the scientific communities and public health communities on these issues. But we also go back and do regulatory science and would go back in phase 4 to do a bioequivalence study to evaluate innovator and generic products again if there were issues that arose. So who does bioanalytical studies? Well, this is a snapshot of the FDA pyramid, and this is kind of a condensed snapshot. But if we go around to that third row, Office of New Drugs, Office of Generics Drugs does bioanalytical studies, my Office of Pharmaceutical Science, and the Office of Translational Science, the Office of Study Integrity and Surveillance. There are other places, but these are primary offices that conduct these studies, and you see at the bottom of the triangle, you see the Division of Product Quality Research, where I'm from. And so how do we do it? I'll give you a snapshot of how bioanalytical studies. I oversee the biopharmaceutics program, and we have a bioanalytical team, we have a chromatography team, we have a rapid response method development team, we have a synthesis team, and we have an advanced analytics team, and we bring these people and these groups together or teams together to address whether it's a preclinical or a clinical study or the complexity of the study, whether it's a simple bioavailability study, or if it's a complex drug device that would be implanted. And we would look not only at local exposure, but systemic exposure over an extended period of time, whether that's 1 day or 30 days. And our goal is to translate CDER's research to support assessment or the review of applications, policy, and our sponsors. We do this in a number of different ways. We do this through guidance support, which you'll see in a few slides. We do it through supporting medical countermeasures, product performance, public health, and increasingly surveillance. So as we look at our guidance, this draft guidance in 1998 or our guidance in 2018, that foundational guidance supported other clinical or in vivo studies, such as the BCS, whether that's both in vitro and in vivo studies, or food effect or excipient effect studies. And when we rolled that guidance out initially, my group actually way back 20 years ago actually rolled this out to the agency in terms of actually being able to develop bioanalytical method that could be used to implement a clinical study to support these types of guidances. And so not only did we support the actual guidance, but we used that foundational bioanalytical method validation guidance to support other in vivo study guidances. We also do, in terms of regulatory support, things like I'd mentioned with excipient effect. And here, what you're looking at is osmotic evaluation of osmotic agents and their impact on BCS class 1 drugs. So sorbitol is an osmotic agent that would remove water from the GI as opposed to a drug trying to transport across the GI. And so for a class 1 drug, high permeability, there's no effect or minimal effect, about 93% of AUCs. But if you look at this in terms of a class 3 drug or low permeability, a significant effect. And so this is important in terms of how you would formulate a product. and we translated that to a guidance, excipient effects, and we published that for our sponsors, for you. Another example is clinical food effects studies. We're looking at class 1 drug. If it's high permeability, will this impact the permeability if you take it with food? And what we found out from those studies that we looked at with metoprolol and propranolol that it didn't. We published this and we translated that into our sponsors. For medical countermeasures, an example of galantamine, which is used in the event, could be used potentially in the event of a nerve agent attack. We developed, because we manufacture in our group, orally dissolving films and transdermals. And from that, we looked at a fast drug release, but we also wanted a prolonged drug release. And we did the studies in animals to be able to show that helping to optimize certain agents will facilitate and support medical countermeasures to protect public health. And we translated that information to the scientific community. We talked a little bit about phase 4 post-market studies. This is bupropion. Study was done in our group, and based on the bioinequivalence, the rating was changed and the generic product was removed from the market. And so finally, public health. Brompheniramine, where we worked with the NIH Pediatric Formulation Initiative to develop formulations that could be used globally, in this case for patient compliance for both brompheniramine and for Tamiflu. And finally, a study for warfarin, where we're looking at different polymorphs. Warfarin has been a concern based potentially on polymorphic forms, and what we showed was that there was no difference in the PK profiles looking at an amorphous and a crystalline polymorph form. And so again, public health. And this information was again translated to the scientific community. As we move to the next part of our talk in terms of advanced bioanalytics, we have set up and we conduct research in a whole series of advanced automation and rapid fire platforms, automation preparative platforms, trying to develop seamless bioanalytical systems and robots that can push a button and the study is conducted. And finally, an example of this is advanced bioanalytics for rapid fire. Real quickly, just in this case, we can analyze samples about 100 times faster than typical analysis by LC-MS, and we believe that this is one of the technologies of the future for advanced bioanalytics. So in summary, the FDA Bioanalytical Method Validation Guidance is a foundational document. The history of the guidance and workshop reports highlights the advancement of the regulatory science of bioanalytical method validation. The FDA works with sponsors, regulators, and academia to translate the regulatory science. And importantly, innovation and the next technological steps for bioanalytics will challenge and will advance the guidance. And finally, I want to thank you for participating. I'd like to acknowledge the workshop organizing committee, my supervisors, Dr. Larry Lee and Dr. Tom O'Connor, to the people in OSIS, Dr. Hadar, Dr. Dasgupta, Dr. Kassim, especially to Dr. Celia Cruz and Dr. Mansur Khan for supporting our efforts in advanced analytics, and to our agency POC, Dr. Brian Booth, who really is a point person for the ICH M10 guidance and the next steps for bioanalytical method validation. Your next presentation that you'll be seeing shortly will be regulatory bioanalysis for large molecules with Dr. Jinwei Zhang.